Development embryology includes differentiation of cells once you start from an egg, and then a reorganization into specific tissues and into the human body. Development can be characterized by different periods of time. You have prenatal development, embryology development, and postnatal development. Okay. Prenatal development. Prenatal development is from conception to delivery. That's prenatal. Okay. That's embryology. Embryology is the development of all your organs in your body during the prenatal period. Postnatal development is development from birth to maturity. So pre-embryonic development is fertilization to implantation. So as the egg travels from the uterine tube to the uterus to implant itself, that's pre-embryonic development. That's when the cells start to divide in the egg, start to form multiple cells that divides. Embryonic development is implantation to the end of the 8th week of pregnancy. Okay, so that's when your embryo develops, from birth to your 8th week. That's your first trimester. I mean, from implantation to the 8th week. It's during the first trimester when everything's developing. Fetal development goes on from the 9th week of pregnancy to birth. So those are different divisions of embryonic cycles, basically. So pre-embryonic, embryonic, and then fetal development. So fertilization. Fertilization is 2 haploid cells that create to form a diploid cell. What's the definition of a haploid? What does haploid mean? Haploid means half the number of chromosomes. A haploid cell has 23 chromosomes for humans. Okay, it's half the number of chromosomes. Remember, sperm and eggs are haploid cells because when they fuse, each one gives you half the number of chromosomes. So then you have 26 total. So the function of haploid cells are the sperm and the egg. The sperm delivers paternal chromosomes, and the ovum provides the maternal chromosomes. Okay, the egg also provides nourishment for the embryonic development. That's why it's larger. It has all the nourishment that the fetus is going to need to develop. Fertilization occurs in the ampulla of the uterine tube. That's in the distal 2/3 of the tube where the swelling is. That's where the large area is. 200 million sperm enter the vaginal canal. About 10,000 make it to the uterine tube. Less than 100 contact the egg, and only 1 will fertilize the egg. So 1 fertilizes it, 100 come in contact with it, 10,000 make it to the uterine tubes, and 200 million are deposited into the vaginal canal. Okay, so the oocyte, which is the egg, oh, there it goes, a little delay, was the egg at ovulation. So when the egg is ovulated, it's surrounded what's called by the corona radiata. Okay, that's what surrounds the egg. It protects the egg as it's being ovulated. Now the sperm to penetrate the egg have an enzyme in the tip, in their cap, in the achromic cap called hyaluronidase. Hyaluronidase acid dissolves the corona radiata protecting the egg and allows the genetic material of the sperm to be deposited into the egg. Okay? So it has to go through that cap using hyaluronic acid. Hyaluronic acid decomposes the corona radiata, so sperm can now enter the egg, okay, and deposit the genetic material, its chromosomes. Okay, this is what an egg looks like, surrounded by about 100 or so sperm. And only one of those is going to penetrate it. Okay, so you have pronucleus formation and amphimixis. The pronuclear material in the egg is the female pronucleus. The nuclear material in the sperm is the male pronucleus. Okay, the amphimixis is the fusion of the 2 pronuclei. The fusion of the male pronucleus and the female pronucleus is your amphimixis. Then this forms a zygote. A zygote is formed when the genetic material of the male and female combine to form the nucleus. Now you have a zygote. A zygote is a fertilized egg. Okay, so here's a zona radii. This protects the egg during ovulation. Okay, this is the sperm that can penetrate here. Hyaluronic acid is released by the chromosomal tip of the sperm. It breaks down the corona radiata, so one sperm can get in inside the egg. Okay, you have the pronucleus of the male and the female. Okay, once they form together, you have an amphimixis. Okay, and a blastomere forms, and you get this genetic material lining up, and you get two. Oops, I'm sorry, we're going back this. I'm going the wrong direction. So anyways, you get these formed, and it comes this way, and you get a amphimixis occurs when the genetic material mixes, and then you start to split the material into two forms, and your cytokinesis begins, where you develop two more daughter cells, and you go to 2, 4, 8, and so on as this develops.
Is it called an amphimixis because it's done in the ampulla?
That's a good question. I don't
Boom.
know. I don't know why it's called an amphimixis because it mixes the two pronuclei together. That's the mixis. I don't know why it's called an amphimixis, but it actually, it very well could be because it occurs in the ampulla of the uterine tubes. It's as good a guess as any. I'll try to look it up. I don't know if anybody knows, but I'll try to look it up and see if I can find out the answer. That's a good question. It very well could be. So prenatal development is known as gestation. Gestation period in humans is 9 months. Prenatal development is divided into trimesters. The trimesters are the first trimester, second trimester, and third trimester. Okay. Usually during the first trimester, you need to be careful. You don't usually do dental work, things like that. Oops, I dropped my pencil. Got it. Okay. That's when everything's developing. You want to avoid X-ray radiation for medical tests, things like that. Second trimester is safe to do most work in. And the third trimester is the female so uncomfortable that you don't want to do anything. Okay. So most of our work and stuff is done during second trimester on pregnant individuals. So first trimester, rudimental, all organs appear. The organs start to develop. Second trimester, it looks like a human. Okay. The third trimester, everything starts to function. The organs, the heart starts to beat. Okay. Stomach starts to move. Everything starts to move around and get rapid growth. So the first trimester, between 1 through 12. Okay, 4 events. Okay, those 4 events occur within the first trimester. You get cleavage. Okay, this is a sequence of cell divisions where blastocyst forms. Cleavage when the cells start to divide, okay, and they start to invaginate in. Okay, you get implantation. Okay, it goes in the endometrial lining of the uterus. That's the inner lining, the functional layer we talked about. Okay. You get placentation, which is the formation of your placenta. So the embryo can receive oxygen, nutrients, and get rid of waste across the placenta. Embryogenesis, development of the embryo. Okay, those 4 things start to occur in the first trimester. So you get cleavage and blastocyst formations. Okay, cell division results in formation of blastomeres. Those are when the 2 cells start to form. Okay. A solid ball of cells develops. That's called the morula. Okay. Cells migrate to one edge of the morula. Okay. Creating a cavity called the blastocele. Okay. The balls of this, the ball of cells is now called a blastocyst, a group of cells. So the outer layer of the blastocyst consists of cells called the trophoblast. That's your outer layer of cells. Okay. So here's your first cleavage. This is entering the uterine tube. This is where fertilization occurs. This is the ampulla, this is the swelling. And then it travels down the uterine tube into the uterus and implants into the wall of the uterus. Okay. So day 0 is fertilization. At day 1, once you fertilize it, you get the zygote. Okay. And then the cells start to divide and you get your first cleavage, which starts to cleave the cells in half. And you see each of the cells starting to develop here. And then you get a 2-cell stage, a 4-cell stage, 8, 16, and so on, until you form what's called a morula. Morula is the ball of cells. Okay. This ball of cells starts to differentiate at about day 6, where you get a trophoblast. Okay. You get a blastocele, and the trophoblast is the outer layer. And this whole thing is called the blastocyst. And this is when this day 6 is when it enters into the uterus. Day 7 to 10, it starts the implantation into the cell and starts to form a placenta where it anchors into the female circulatory system. Okay, so the trophoblasts were on the outside. They're going to provide the nutrients to the developing embryo. Those are the outside cells of the blastocyst. The inner cells of those are your stem cells. That's going to develop all the cells of your body. That's going to differentiate into an entire fetus. Okay, so when it contacts the endometrial lining, okay, the trophoblast cells start to divide rapidly. They fuse with the endometrial lining, forming what's called a syncytial trophoblast. Okay, the layer cells release high hyaluronidase, or the hyaluronic acid, that erodes away the endometrial lining, so the mass can implant there. Okay. So hyaluronic acid is used for the sperm to penetrate the corona radiata of the egg and cause fertilization. And hyaluronic acid is also used it's released by the trophoblast layer to eat away some of the endometrial lining so the egg can implant into there more successfully. So 2 uses for hyaluronic acid. Upon implantation, when the egg implants into the side of the uterus, the inner cell mass separates from the trophoblast area. Remember, the trophoblasts are the outside cells that are anchoring into the uterus, and then the inner cell mass is going to form the fetus. When the inner cell mass separates, 2 cavities are formed: the amniotic cavity and the blastocele cavity. Okay, so here's your blastocyst. Okay, remember your outer cells, your trophoblasts, they release hyaluronic acid and start to dissolve part of the uterine wall here, the endometrial lining. The egg starts to implant, and once it implants, this blastocele starts to form 2 layers. They form amniotic cavity, okay, and they form a blastodisc. Okay, once the blastodisc is formed, the layers of cells form the amniotic cavity and the blastocele cavity. The layers are called the epiblast and the hypoblast. Okay, those are layers of the form between the amniotic cavity and the blastocele cavity. So you have an epiblast and a hypoblast. Okay, so take a good look at it. Here's your blastocele. Okay. This is the syncytial trophoblast. It's the outside layer anchoring into the uterus. The cellular trophoblast is your outside cells. Here's your amniotic cavity, which is forming. This is the epiblast that forms the cells right around the amniotic cavity. And this is the hypoblast. Remember, this forms between the inner mass and the trophoblast cells, your amniotic and the amniotic cavity, your epiblast and your hypoblast. Okay, gastration and the germ layer formation. Some of the cells of the epiblast move towards the center of the blastodisc. That creates what's called your primitive streak. Okay, this is called gastrulation. As the cells move towards the primitive streak, they begin to migrate between the epiblast and the hypoblast. That creates a 3rd layer. Okay, and the 3rd layers of the gastrulation are the ectoderm, which is derived from the epiblast layer, the mesoderm, which is a new layer between the epiblast and the hypoblast, and the endoderm, which is from your hypoblast. So the new layer formed between the epiblast and the hypoblast is what's going to be your mesoderm. Your epiblast becomes your ectoderm, your hypoblast becomes your endoderm. And those are your 3 germ layers right there: ectoderm, mesoderm, and endoderm. Okay, this is same thing we just did, how it happens. The pronucleus forms and the cytokinesis forms. It's just a summary of exactly the events that occurred as we talked about. Okay, so the ectoderm. Ectoderm is your integumentary system, your skin, your skeletal system, your nervous system, your endocrine, respiratory, and digestive system. That's your ectoderm. Okay, the mesoderm also contributes to your integumentary system, your skeletal system, muscular system, etc. Same thing as before. And with the endoderm, endocrine system, respiratory system, digestive system, urinary and reproductive. Okay, those are your 3 germ layers: your ectoderm, your mesoderm, and your endodermal layer. Okay. So the extraembryonic membranes, the ectoderm, mesoderm, and endoderm, okay, are your germ layers. Each layer will form specific tissues and organs of the body. Okay. There also form structures involved in embryonic survival called extraembryonic membranes to help keep the fetus developing. Okay. The extraembryonic membranes, there's 4 major extraembryonic membranes that help to keep the fetus going. You have a yolk sac, an amnion, an allantois, and a chorion. Those are the 4 different extraembryonic membranes that the fetus is developing. Okay. So this shows you as your development here, you get migration of the mesoderm on the inner surface of the trophoblast, and you see where your amnion is starting to develop. This is a syncytiotrophoblasts that anchor it to the uterus. You get your chorion, which is part of your mesoderm and your cellular blast. You still have the yolk sac developing, and you have the inner layer, that space is the blastocele. That's at week 2. At week 3, you see some of the differences developing. You can see your extraembryonic membranes. There's 4 of them, remember? There's the amnion, allantois, yolk sac, and chorion. You can see how those start to develop. Okay. The yolk sac. What the yolk sac is going to do, it extends from the hypoblast cells into the blastocele. It's the early site of blood cell formation, is your yolk sac. The amnion is amniotic fluid, fills your amniotic cavity. This surrounds the fetus with a liquid and helps protects the fetus and cushions it from any movements. That's your amnion. Allantois gives rise to your urinary bladder. Okay, and the chorion. This is your mesoderm and your trophoblast form the chorion. And the chorion develops extensions into the endometrium and anchor the eggs, into the fertilized eggs, into the uterus. Placentation begins to form when the chorion, okay, produces your villi. With the chorionic villi, they extend into the endometrial lining. So those little fingers extend to it and they form or fuse into the endometrial lining, allows nutrients to get into the egg cells. Now the body stalk connects the embryo to the chorion. Okay, so as the fetus develops, gets larger and larger, it moves farther into the uterus, gets impacted into the wall, okay, into the endometrial lining, the functional layer of the endometrial lining. And it gets its nutrients from the umbilical cord. That's where the fetus gets all its nutrients from. Okay. You can see it developing into the wall of the uterus here. You see you have the uterus, the myometrium. The myometrium is the muscle part of the uterus. The endometrium is the inner lining, and the perimetrium is the outer lining. Remember those three layers of the uterus. Okay. You can see the yolk sac real clear. This is the chorion villi of the placenta. You see how they're invading. All these little villi are invading into the uterine tissue. Okay. So you get placental circulation. So blood flows from the fetus to the placenta in paired umbilical arteries. Okay, remember, arteries flow what direction? They flow away from the heart, right? Okay, so the umbilical arteries flow from the fetus's heart back towards the maternal blood supply, back towards the placenta. So you have 2 paired umbilical arteries, and then blood returns a single umbilical vein back to the heart of the fetus. Okay. The umbilical vein is oxygenated blood because it's coming from the maternal arteries. Okay. And the vein flows back to the fetal heart. So it's a vein, but it's deoxygenated blood. It's oxygenated blood. The arteries are deoxygenated blood. They're flowing back from the heart out back towards the placenta. Okay. So that's how the circulation system works in the fetus. You can see the umbilical cord, and this whole area here is the placenta. Okay, where the chorion has divided into the uterine lining. Okay, then you got your amniotic cavity surrounding the fetus to protect it, which is filled with amniotic fluid. Okay, Fiber optic human development. This is 8 weeks old. You can see the amnion sac filled with amniotic fluid that's protecting the fetus. You can see the placenta, which the umbilical cord is embedded into. And you see the chorionic villi that extend into the uterine wall. Okay. Another view of it. There's the different layers of it. Okay, you're in the uterine cavity, in the uterus. This is the endometrial lining on the inside. Here's your amnion with amniotic fluid all filled in here. It helps protect the developing fetus. Okay, you have your placenta, which is the area that's embedded into the uterus with the chorionic villi. And you see your umbilical arteries. Remember, there's 2 umbilical arteries. There's a paired set, one right and one left. And blood is flowing away from the fetal heart through the umbilical cord. And that is deoxygenated blood. The blood's already being used by the fetus. Okay. You have your umbilical vein. There's only one, and it flows towards the fetus, into the heart. Remember, veins return blood to the heart. This is oxygenated blood. This is coming from the maternal source, and this is your oxygenated blood. Okay. That's what we need. Okay. So embryogenesis, okay, development of the embryo. Shortly after gastrulation, embryogenesis begins to start. By week 4, a head fold and a tail fold starts to develop. The embryo is separated from the blastodisc area, starts to form on its own. Okay, you see the different outlines of it. You can see the extra, remember there's 4 extra embryonic members you talked about. There's the amnion, the allantois, the yolk sac, and the chorion as they develop. Okay, that's week 3. As week 4 progresses, you see how the yolk sac starts to extend, how the chorion starts to extend into the uterus to make bindings into that tissue. You get embryonic head fold, and the embryonic gut starts to form in your fetus at week 4. At about week 12, your organs begin to form. Heart, the lungs, kidneys, etc. That's called organogenesis. Organogenesis is when the development of the organs start to form about week 12. Okay. This is human development. This is again at week 4. You can see the development, you see the body size, you see the tail, you can see the legs starting to form, the arms starting to form. So much for your nervous system, your central nervous system, the pharyngeal arches start to form. You can see where there's ear, medulla, forebrain, all that's starting to form at week 4. Second trimester, third trimesters. The second trimester, the fetus is covered by the amnion. The amniotic fluid surrounds it. Fetus grows faster than the placenta as it starts to expand in size. In the third trimester, all your fetal organs become functional. So once the third trimester becomes, all your organs start to work. Okay, that's a 4-month-old fetus seen through a fiber optic endoscope. Okay, see how it's pretty much fully developed, and it just has to grow. Okay. Changes in the uterus during gestation. The uterus is going to increase from about 7.5 centimeters to 30 centimeters. That's over a 4-time increase in size as the fetus develops. Contains almost 5 liters of fluid. That's the total amount of blood you have in your body is roughly 5 liters. So the uterus alone contains 5 liters of fluid surrounding that fetus. The uterus and the contents within it, within the fetus, weigh about 22 pounds. Okay. Abdominal organs are pushed out of the way of the normal position as the fetus grows. Okay, here you have a view of the developing fetus. You can see the uterus, the uterine wall, the amniotic fluid which surrounds the infant. The cervix is the bottom tip of the uterus where the baby's going to come out from the birth canal. This is at 16 weeks development. You have the umbilical cord that connects the fetus to the maternal blood supply. And you have the placenta, which is embedded into the wall of the uterus by the choroid villi. Okay. As after dropping, the baby moves its position. So as it grows, you see 3, 4, 5, 6, 7, 8, 9 months. Then it starts to drop as it gets ready for delivery. The head comes down to the birth canal, and the baby starts to drop, and then the woman starts to carry the baby lower. Okay. So you can see when the baby develops, the head comes down. This is going to be the birth canal here. This is the cervix of the uterus. And as the baby pushes, that starts to dilate. Right? You can see as the baby's developing, everything gets squished, especially look at the urinary bladder. Urinary bladder right there. You can see how that urinary bladder has to get less and less, be able to hold less and less as it's getting squished there. And the stomach, everything else gets pushed out of position as the fetus grows. Okay. The goal of labor is parturition, is birth. Okay. When you separate the baby from the mother, there's 3 stages of labor. There's dilation, there's expulsion when the baby comes out, and there's a placental stage where the placenta comes out. Dilation stage. In dilation, fetus moves down the cervical canal. Okay, the cervix starts to dilate. The amnion fluid ruptures, and basically water breaks. Okay, the expulsion stage. Expulsion is birth of the child. Placental stage is ejection of the placenta after the baby's born. Okay, those are your 3 stages of labor. So you got dilation of the cervix, expulsion, which is birth, and then placental, which is ejection of the placenta. Okay. So this is the dilation stage. Okay, see the baby pushing on the cervical area of the uterus. It starts to open up, and that's dilation. Expulsion stage, when the head crowns, the baby starts to come out. And then after the baby's born, you get ejection of the placenta. 3 stages of labor and delivery, 3 stages of labor. Okay, these just exaggerated view of it. Again, you can see the stage 1, dilation, stage 2, expulsion. You can see the cervix is completely open here for the birth canal. And then you have the placental stage where the placenta is ejected after birth. Okay. Premature labor is labor that begins before the fetus has completed normal development. The line between spontaneous abortion and immature delivery is usually set at about 17.6 ounces. Okay, so a little over 1 pound, and let's see, 1 pound is 16 ounces. So it's 1 pound, 10, so just under 2 pounds. Kind of between 1 and 2 pounds is the limit they set for when the baby's viable or can be resuscitated. And basically it's because the respiratory, this doesn't have development to support life. Most of it is in surfactant. Remember we talked about surfactant in the lungs? Surfactant coats the lungs so they don't collapse every time you take a breath. When the baby's that little, they have no surfactant. So every time the baby tries to take a breath, the lungs completely collapse. You got to re-expand those lungs every time. And that's not compatible with life. So they usually set about 17.6 ounces. So right between 1 and 2 pounds is what they usually use to determine if the baby is survivable. So premature labor on the neonatal period is from birth to 1 month. That's your neonatal period. Okay. During this time when you're born, the lungs fill with air. Because remember, while the baby's in the uterus, the lungs are not functioning. Okay, they're filled with fluid. Okay, once the baby's born, the lungs fill with air, the baby starts to breathe on their own. Blood circulation changes with the closing of the ductus arteriosus and the foraminal valley. We talked about that because blood is shunted, okay, away from the lungs, away from the pulmonary trunks. Because there's no good to have the blood go to the lungs because the lungs don't have any oxygen there. So if a baby's in utero, okay, there is no blood going to the lungs. It's being shunted by those 2 openings, foraminal valley and the ductus arteriosus. Okay. Heart rate then starts to drop from 120 to 140 beats per minute to about 70 beats per minute. Okay. And breathing rate goes from 30 a minute to normal. Normal is about 6 beats a minute, 6 breaths a minute. Okay. The kidneys start to filter the infant's own blood. Okay, instead of using the placenta from the mother, now the kidneys start to work. Digestive system starts to become active in that first month. And the metabolic rate is increased to maintain warmth a few days after birth. Okay. That all happens in the neonatal period once the baby's born. So we're going to go over all we go through this chapter, through all the development of the embryology of each of the different systems for development. The integumentary, which is our skin system, the skull, vertebral column, appendicular skeleton, muscles, nervous system, spinal cord, brain, special sense organs we talked about, endocrine, heart, cardiovascular, lymphatic, respiratory, digestive, urinary, and reproductive system. Those are all systems we covered already, and we just kind of review how they develop in embryology, embryologically. Integumentary system, your skin. It's the epithelium overlies the mesenchyme. That's your integumentary system. That starts to happen about 1 month. About 3 months, epithelium multiplies to form the different layers. Remember thick skin and thin skin and the different layers that we have in the integumentary system. Okay. The mesenchyme becomes dermal connective tissue. After 4 months, epidermis grows into the dermis, okay, forming columns of cells, which becomes your hair follicles, your sweat glands, and your sebaceous glands. Those are on the integumentary system. About 4 months, those start to form. So here's your ectoderm, which is going to form your epidermis, and then your mesoderm underneath it. As things develop, you start to form layers here. Your epidermis starts to form layers, and it starts to invaginate. This forms your glands. Okay. The skin at about 4 months, epithelium continues to thicken. Melanocytes migrate to the stratum basale layer. Remember, the melanocytes are found in the stratum basale layer. What do melanocytes do? Melanocytes control what?
Pigment.
Pigment, skin color. If a person is an albino, do they have the same amount of melanocytes as a person who's not an albino?
Yes.
Yes, they do. That'll be on your nursing test. Okay. Then melanocytes just don't function correctly. They don't produce melanin. Okay. But they have the same number of melanocytes as everybody else. Then you got the nails start to develop from the thickening of the epidermis. You start your nail beds start to form, your hair starts to form, and your glands. Okay. You can see the nail field here as this starts to thicken. Okay. It starts to develop your nails, your fingernails, and your toenails. Okay. You can see the melanocytes starting to form and move into the stratum basale layer. Okay. We got your epidermis, your dermis, and your hypodermis. Okay. Remember, the dermis has two types of tissue: loose connective tissue and dense connective tissue. All right, hair follicles start to develop. They develop as deep column of cells surrounded by a papilla. Okay. The exocrine glands develop as your epithelial columns elongate and start to coil around. Remember, the ducts of exocrine glands are coiled. And mammary glands start to develop from the thickening of the epidermis. So you got your hair follicles, your exocrine glands, and your mammary glands start to develop. Exocrine release its contents where?
Outwards, out of the body.
Out from the body, on the surface of the epidermis. Okay. Your endocrine glands release it into the body, into the lumens of blood vessels or lymphatic systems or lymphatic vessels. Okay. Again, hair follicles start to form. Okay. This is your papilla here. It starts to form an invagination of the epidermal tissues. And this muscle here is responsible for pulling that up. What's the name of this muscle here? It's responsible for goosebumps as your hair on your surface of your skin.
Something apparatus.
How about the erector pili muscle?
Ah!
That's your erector pili muscles, responsible for your goosebumps as it develops around your hair shafts. Remember, you had coils, a sweat gland, the duct's coil, and it's an endocrine gland. It releases its contents to the external surface of the body. You can see how it goes from 5 months to birth. As the colon starts to develop, and it starts to become a continuous tube for the gland to release its contents. Again, you see the ducts that are branching. The epidermis starts to invaginate into the dermis, and this starts to form your duct system. Okay, these are your mammary glands that are starting to form. Development of the skull. We talked about that a lot before. We talked about all the bones, development, how the bones don't fuse in the skull, so the baby can fit on the birth canal, the skull can squish. So 5 to 8 weeks of development of the skull after fertilization cartilage begins to appear. Cartilage enlarges the brain area to form condocranium. Cranium forms as a cartilage around the brain. Skull walls and floors are beginning, but no roof is attached yet. No roof starts to form. Endochondral ossification appears. Frontal and parietal structures appear. The frontal bone, parietal bones are on the side. Okay, they start to appear at week 9. Okay, you can see the skull developing. You can see development of the skull. This is the eyes, the brain, the pharyngeal cartilage. This is where your skull is going to develop right here in this area. That's 5 weeks. At 8 weeks, that's a little more development. You see some of the cartilage starting to form. You see the condocranium. This is the cartilage going to case the brain up here as it starts to develop. You see your spinal column starting to develop, your mandible here. You got your nasal cavity, your eyes. This is the part of the brain that starts to develop here. Okay, as that develops, you got your frontal bone. Remember, this is going to contain the frontal area. The side is going to be your parietal bone. It's going to develop. This is your occipital bone. Remember, the frontal bone is the most anterior bone of the cranium. The occipital bone is the most posterior bone of your cranium. Temporal bones and parietal bones lie along the side. You can see the mandible and the maxilla starting to develop here too. Starting to fill in, become ossification. Okay. See your hyoid bone starting to develop. Your larynx, trachea. All starting to develop. This is at week 9. Okay. At week 10, the mandibular arch is fused with the condocranium, and then the hyoid starts to form. At week 12, ossification is well underway. So bone is starting to replace the cartilage in the cranium area. Okay. At birth, the cranial roof remains incomplete. The cranium is not fused. Remember, we talked about that. Those are your fontanelles, your anterior fontanelle and your posterior fontanelle. That's so when the baby goes down the birth canal, the head can deform. It can squish down. You can't bend bone, but you can bend cartilage. So that the cartilage can bend and the baby's head can distort as it goes down, makes its way down the birth canal. Okay. So your anterior and posterior fontanelles are present at birth. So the skull is not completely calcified, it's not completely ossified at birth. Okay, so at 10 weeks, we saw the frontal bone. Now we got the parietal bone here. You got your occipital bone developing in the back. You got your frontal bone. Okay, you got your maxilla and mandibular arches starting to form, and your hyoid starting to form. Okay. At 12 weeks, you have a lot more bone growth. You can see the zygomatic arch, which is the side of your cheekbones, right? So your mandible, your maxilla, the frontal bone is becoming extremely large here. Your parietal bone is becoming large. This area here is not going to fuse. That's your anterior fontanelle, and this will be your posterior fontanelle. Remember, the fontanelles don't fuse, so the skull can be deformed. And here's a real good picture. This is at birth. So the skull is not, or the cranium, sorry, is not completely fused with bone at birth. You got your fontanelles and you got your sutures that have not fused yet. But you can still see your occipital bone, your parietal bone, your frontal bone, mandible, maxilla, nasal bone, etc., all formed. Okay, the vertebral column. Okay, the vertebral column, 4 weeks on either side of the developing spinal cords are blocks of mesenchymal tissue called somites. That's going to form on the developing spinal cord. The medial side of the somites, the sides that are closest to the middle, when they start to develop, are going to form your vertebral column. Okay, those somites, the middle portion of that are closest in towards that center is what's going to form the vertebral column. The vertebral cartilage forms around the spinal cord. The sacrum and the coccyx become fused together as this develops at week 8. Okay, so this is a follicle. Here's your spinal cord. Here's your somites. This tissue, this mesenchymal tissue, the medial portion right here, this inner portion, this is what's going to form your vertebral column. Okay, the spinal cord starts to form. This is mesenchymal with the somites here. Again, starting to develop these mesial portions of it. From 4 weeks to 6 weeks, you can see how they're developing into the bone starting to ossify. And in between this, we have the cartilaginous pads. Okay, what fills cartilaginous pads? What fluid? The trick question.
None.
That's right. There is no fluid. Cartilaginous pads, most nursing exams A lot of students, I got the results from the nursing exams from 2 years ago. And most students put cartilaginous pads are filled with synovial fluid, and they are not. Synovial fluid fills your TMJ, your elbow joints, your knee joints. That's synovial fluid. Okay. Your cartilaginous pads are just cartilage. And all they do is prevent compression of your vertebrae when you run, when you jump, things like that. Okay. So no fluid fills the cartilaginous pad. Don't miss it on your nursing exams. Okay. They're just they're cartilage. It's a cartilaginous pad. Okay. And again, as you develop 8 weeks as an adult, you can see your vertebrae develop with your transverse processes here. And you can see in between each one of these is your intervertebral disc. The intervertebral disc is a cartilaginous pad. Okay. At week 12, your ribs and your sternum start to do ossification. They start to form bone. Okay. The tips of the longer ribs do not ossify. They remain as cartilage, form the costal cartilage all through your adult life. So the tips of the long ribs are costal cartilage, are not bone. Okay. So here's your spinal cord development again. There's the transverse process you can see developing. Okay, and it's your spinal cord developing in the center here as it ossifies around it. And the baby is surrendering, I guess. I know it gives up 8 weeks to 9 weeks. You can see it from just 1 week. You can see how this the sternum has fused together. Let's come together. You get the manubrium, the body, and this is going to form your xiphoid process right here. And the ribs are starting to form their construction across there. The appendicular skeleton. Appendicular skeleton is what? What's appendicular mean?
Appendages, arms and legs.
Right, the limbs, upper limbs and lower limbs. The axial is your center. The appendicular is your arms and the appendages that hang off the axial, which is your arms and your legs. Okay. At 4 weeks, ridges start to appear along the length of the embryo. Okay. These ridges accumulate at the superior end and the inferior end. They form your limb buds. They form the end of the limbs and the beginning of the limbs. At 5 weeks, the limb buds start to form cartilaginous tissue. Okay, and these models develop between 5 to 8 weeks. They start to form fingers. They start to form hands from just a paddle shape they start out as. So 5.5 to 8 weeks of the upper limbs. Bends develop at the shoulder and the elbow. Okay, lateral rotation of the apical ridge occurs to create the correct position for the elbow. So the elbow starts to align itself as it rotates into position. Hands originate as paddles. Remember, they form as paddles at first and eventually form fingers. So at first they'll form paddle shape. They'll form your fingers like this, and then you'll digest the cell that eat away this tissue that's formed and start to develop your digits. So it forms a paddle at first. Okay, so surfaces in those joints remain covered with cartilage. On your lower limb, the bends develop at your hip and your knee. And then medial rotation of the apical ridge occurs to create the correct position for your knee. Feet also originate as paddles, but eventually individual toes form as the body digests the tissue between the toes. Same thing with the fingers. So surfaces where the joint remain covered with cartilage also, just like in the upper limbs. Okay, and this is how it forms. You see how this paddle forms for your hand, and the bone starts to develop. And then this tissue is eaten away and starts to form individual fingers as the bones develop on your digits. But the original thing, like 5.5 weeks or so, you have a paddle that forms there. And then we digest it and break away, eat away the tissue between the fingers. That's why some babies are born with webbed hands and webbed feet. They haven't broken down the tissue between the fingers or the toes yet. That's what happens. Okay. This is your appendicular skeleton again. Again, you can see your hand developing. You have a paddle there. That's as you're 5.5 to 7 weeks. Okay, you got your joints that start to rotate. Okay, you get your cartilage that starts to fill into the joints, and then that forms the joint cavity. Appendicular skeleton at 10 weeks continues the ossification, continues bone growth. Distal bones of the carpus and the tarsus, and the wrist and the foot remain cartilaginous. Consists of, at birth, you still have extensive areas of cartilage. Okay, all the bone is not covered in it when you were born. You're still fine. Babies are very flexible. That's because a lot of the bones haven't developed and there's a lot of areas of cartilage. Okay, so birth to 10 weeks. I'm sorry, I'm looking back. Yeah, from 10 weeks to birth. You can see all the blue is cartilage. Okay. You see how much that has filled in the bone. We still have cartilage left. Okay, and a lot of it is in your joints. You ever look at a baby that you can take their legs and go, because there's no joints yet. It's all cartilage. They've been in tremendous flexible positions. Okay, that's because there's still a lot of cartilage, especially in the joints. But you can see all the ossification has occurred from 10 weeks to birth. Muscles. At 4 weeks, the mesoderm on either side of the notochord forms the somites. The medial portion of the somite forms your skeletal muscle called the myotome. Okay, it forms your spinal cord, and around the notochord, it forms your skeletal muscle, called your myotome. Myotome areas develop in posterior muscles. You have epaxial muscles, okay, and you have hypaxial muscles, okay. Anterior, those are posterior muscles. Epaxial are posterior. Hypaxial muscles are anterior muscles that form. Okay, here's the somites that are forming. This is where the lateral plate is going to form. That's where your muscles are going to start to develop. Okay, you can see the different muscles form on the sides on the anterior sections and posterior sections. You're going to start to form extensor muscles and flexor muscles. Remember, extensor muscles, okay, move it creates an angle between the two bones as it creates a greater angle as it contracts. Okay. Flexor muscles decrease the angle between the two bones. So you take your arm and you bring your arm together towards your shoulder, that decreases the angle of the elbow. Those are flexor muscles. As you pull your arm away from your shoulder, those are extensor muscles. They increase the angle of the joint at the elbow. Okay. So your epaxial muscles and your hypaxial muscles you can see developing here. They also got your lungs and your ribs starting to develop with that. At 7 weeks, muscles of the face, the back, and abdomen start to develop. At 8 weeks, your limbs enlarge and muscles continue to develop. Okay, so another diagram showing the exact same thing. You have your extensor muscles and your flexor muscles developing. Okay, at 8 weeks, you can see your your flexor muscles and your extensor muscles, your flexor muscles and your extension muscles as they start to develop. Okay, at birth, rotation of the upper limb and lower limb creates muscles of flexion and extension we just talked about. Those are your flexors. Again, flexors decrease the angle between the joint. Extensors increase the angle between the joint. So your anterior muscles of your arm are flexors. Your anterior muscles of your leg are extensors. The posterior muscles of your arm are extensors. The posterior muscles of your legs are flexors. And development of the nervous system. We covered muscles, appendicular system, now go on the nervous system. 20 to 23 days, so 3 weeks out. Somites appear on either side of the notochord. The somites appearing in the mesial aspect form your vertebral column. Somites on the lateral side forms your muscles, the flexors and extensors. So a neural plate is formed. So a crease developed on the length of the notochord. Okay. The edges of this crease are your neural folds. The neural folds form your neural tube. Okay. The neural tube becomes your central nervous system. That's what forms your nervous system. So axons, okay, extending from the neural tube becomes your peripheral nervous system. Okay, so the tube starts to fold from these neural folds form a tube. This tube forms your central nervous system. What branches out from there, your axons, is your peripheral nervous system. Okay, so here's the neural plate that starts to form and starts to come up and starts to form a fold, and it folds over. And this forms your neural tube. Okay, these neural folds form your central nervous system, and the axons come out from that, form your peripheral nervous system. Okay. You can see the neural cell inside of here. You can see how the ependymal cells line the layer here. And how's it going to form your central nervous system? When the neurons kind of come out, that forms your peripheral nervous system. Remember, astrocytes, oligodendrocytes, those are supporting cells in the central nervous system to help maintain the neurons. Okay. So the spinal cord, the neural tube is closed. It produces 3 layers. We saw the epidermal layer, mantle layer, and the marginal layer. Okay. Those are 3 layers of your spinal cord. Neurons start to develop in the mantle layer, the middle layer. That's where your neurons start to develop. Axons grow from the mantle layer to the marginal layer. They start to work their way outwards away from the tube. Okay. Axons form your bundles and tracts in the spinal cord. Remember all the bundles that run the different tracks that we have that connect up to the thalamus and branch up into the cerebral cortex. Okay. So there's your neural tube. These are the 3 layers. Your epidermal layer on the inside, your mantle layer on the middle, and your marginal layer on the outside. And as the nerves grow, they grow in the mantle layer, the neurons, and they extend out towards the marginal layer as they develop embryologically. Okay. At 7 weeks, remember, ventral and dorsal roots start to develop. They come in, you have your spinal cord in the center here, and you got ventral roots coming in, you got dorsal roots. Okay. The glial cells start to develop. Remember, glial cells are non-neuron cells. They do not carry an impulse. The function of glial cells is to support and maintain the health of the axons, the nerve cells. Okay? Spinal and cranial meninges develop, and then your spinal nerves develop. Okay? The cranial meninges, how many meninges are there developed in the cranium? How many layers?
3.
3. What's the innermost layer? The thin layer. When it looks like cellophane on the brain. It's just really thin, protects the brain.
Dura? No.
Pia mater. Then the middle layer is your arachnoid layer. And the out one is the dura. Man, that's the real thick one on the outside. Those are the 3 meninges that start to develop. Your pia layer, your arachnoid layer, and your dura mater. Right? And this is at 7 weeks. At 7 weeks, you can see developing of the, the dorsal roots. They start to develop. You start to see the development of the larynx. You see the teeth developing along with the mouth going to form. You have your dorsal root ganglia. You have the suprarenal medulla, which starts to develop above the kidneys, where the kidneys forms. Your meninges, which form your 3 layers, your pia mater, your arachnoid mater, and your dura mater. You have the autonomic ganglia are developed, and melanocytes, okay, for pigmentation. That's all at 7 weeks. And a lot of this comes together. You get very hard to isolate everything. A lot of things are developing at the same time. Okay. And it's at 7 weeks, you can also see the cranial nerves and ganglions start to develop. Okay. Remember your cervical plexus and brachial plexus in the arm as a nerve exit from the central nervous system to the peripheral nervous system. You got your spinal nerves that come out, lumbosacral plexus down in here. Okay. Developmental abnormalities. When the nerve system is out, you can have abnormalities. Spinal bifida we talked about. Spinal bifida is when the nerve tube doesn't completely cover all the way over the nerves in front of the canal they go through. There's an opening, and some of the nerves can come out. So in spinal bifida, the vertebral laminae fail to unite. So it does not seal that tube. So your neural arch is incomplete. Okay. So in spinal meninges, those meninges layer that are cases around the spinal cord, the 3 layers, the dura mater, the arachnoid mater, and the pia mater, with the nerves inside, they tend to bulge out of the spinal cord and make a bulge outside the body. Okay. And depending how big it is and what nerves it affects, can cause paralysis, can cause all the kinds of things at birth. Okay. That's spina bifida. It's failure for the nerve tube to fuse completely. You can have a neural tube defect where a portion develops as a flat plate instead of a round tube. Part of this comes in as a flat plate across there. That's a neural tube and puts pressure on those nerves. Okay, so this is spina bifida, where the nerves actually come out of the body. The neural tube defect is the exact opposite, where everything's evaginate in. There's a flat area where it doesn't come out and form that full circular tube around it. So you got a flat area back in there. Okay. Development of the brain. Okay. At about 23 days, so it's a little over 7 weeks, the cephalic extension is one of the 3 brain vesicles: the prosencephalon, the mesencephalon, and the rhombencephalon. The prosencephalon forms your forebrain, mesencephalon is your midbrain, rhombencephalon is your hindbrain. Okay, this is the development of the brain. You see the 3 as the arch forms. You see this is the neurocele, the covering over it. Okay, you see your prosencephalon, it's about your frontal lobe. Mesencephalon, this is your medulla oblongata, your pons, and then your rhombencephalon, this is your brain stem coming down. So at 4 weeks, the rhombencephalon, okay, divides into the metencephalon and the myelencephalon. Okay, the prosencephalon divides into the telencephalon and the diencephalon. Remember, the mesencephalon does not divide. That forms your midbrain. That one does not divide. Okay. So again, here's the prosencephalon, forming your diencephalon, your telencephalon. Here's your mesencephalon, which does not divide. It's one that doesn't. And then your rhombencephalon forms your metencephalon and your myelencephalon and feeds into the spinal cord. Okay. At 5 weeks, the mesencephalon midbrain develops. Cranial nerves begin to develop. At 8 weeks, the choroid plexus starts to develop, and you start to form your cerebral hemispheres in the brain. At 11 weeks, the cerebellum starts to develop. Remember, the cerebellum is responsible for coordinating your movements. Okay? It feeds information from the brain and allows you to move in a coordinated manner, so things are not jerky. Okay, so at 11 weeks, your cerebellum is actually starting to develop. Okay, you can get the cranial nerves starting to develop. Cranial nerve number 3, 4, 5, 7. These are cranial nerves that are starting to develop here. There's 9. There's the vagus nerve number 10, number 12. Okay, all those starting to develop. Yeah. Okay. Again, another view of the same thing there. 8 weeks, you can see the flexure as it comes down, and you see your cranial nerves all starting to develop in those areas. Okay. This is your cerebral hemispheres of a child. This is the cerebellum, the cerebrum on top. This is the medulla oblongata and your cranial nerve number 11 coming down. And this is the spinal column right here. You see development 11 weeks. You can see how this is developing. Okay, this is your diencephalon and your telencephalon area. Okay, this is the mesencephalon, midbrain, and this is your cerebrum and your pons down below that. Oops, wrong button. Okay. Vision, how your eyes develop at 4 weeks. Your optic vesicles. Then is your optical nerve is cranial nerve number 2. Okay, that's the optic nerve. Okay. You have the optic chiasm, so that means your right eye information goes to the left side of your brain. Your left eye information coming in goes to the right side of your brain because the optic chiasm, those nerves cross at the optic chiasm. The bulge edentulous forms your optic cup as the eye starts to develop. Okay. The epidermis of that cup forms the lens of your eye. Okay. So as your eye develops, it starts to form what's called a primordial cup or an optic cup right there. Okay. As that cup starts to develop, it pinches off here, and this forms the lens of the eye. Okay. Olfaction. Olfaction is cranial nerve number 1. Okay, that's your sense of smell. You get receptors that extend along the base of the cerebrum and end in little bulbs. So they come on the base of the cerebrum and they'll form bulbs and extend back down. Okay, that's your olfactory nerves. These are receptors. Remember, they go through the cribriform plate of the sphenoid bone, all those little holes, receptors, and give you the sense of smell. Okay, you can see at 5 weeks, you can see the eyes are developing and the nasal plaque is starting to develop. And by 10 weeks, you can see the external nares have actually developed. Okay, gestation. Taste. At 5 weeks, taste buds develop. These nerves go into the mouth, okay, and your pharynx area. That's where your taste is. Okay, taste along the roof of your mouth and along your tongue. When nerves innervate the tongue, epithelial cells differentiate to form your gustatory cells, which are your taste cells. Okay, here's your gustatory cells right here as they develop. Okay, this is a sensory neuron. This is the epithelial cells of your tongue. Okay, and as they form, these gustatory receptors bind to the taste buds. So that information can be centered back towards the brain so you can determine what you're tasting. Okay. Hearing. Hearing is equilibrium in sound. At 3 weeks, otic plaques start to appear on the lateral sides of the rhombencephalon. Okay. Creating otic vesicles. The vesicles form the bony labyrinth of your ears. Okay. You can see how the otic plaques start out from a neural groove and start to form over. And then you start your development of your bony labyrinth here that's going to develop into the ear. Okay. And then at 7 weeks, you see what it starts to look like. As your vestibular ganglion, everything starts to form, you're developing ossicles. Okay, these are your developing ossicles right here. Those are the 3 bones. And you can see the 3 bones here is ossified. And those are your auditory ossicles, the smallest bones of the human body. They are the malleus, the incus, and the stapes. And those are the 3 auditory ossicles of the ear. On the practicum, if you see a picture like this, and you see an arrow pointing like that, do not put ossicle bones, auditory ossicle bone. That is the stapes of the ear. Be specific what I'm pointing to. Okay. Know what it's pointing to. Then this is the temporal bone here. Your tympanic membrane is right there. Okay, that's your tympanic membrane. That's your eardrum. Your external acoustic meatus is your opening to the ear. Your auricle is the soft tissue of your ear. And those are the 3 ossicles. Remember, the stapes, the incus, and the malleus are the 3 auditory ossicle bones of the ear. Endocrine system. All the hormones are in that develop. We got several pharyngeal pouches start to form. Let me see if I play that. Okay, they form your thymus gland, your parathyroid gland, and your thyroid gland. Okay. Which one of those 3 is 4 little purplish dots on the back of one of the glands?
Your parathyroid.
Parathyroid. Your parathyroid is to have the right and left side and 2 dots on the back of the thyroid gland. That's your parathyroid. Okay, you see the parathyroid is developing here. You got thyroid gland developing. And look at the posterior side, you can see each of those dots on the parathyroid developing. Okay, your thyroid gland develops, comes out from pocketing, forms an invagination of epithelial cells, and then forms a thyroid gland. Okay, again, the pituitary gland. Okay, there's a bony part of the skull, part of the sphenoid bone, where the pituitary gland fits into. What's this part of the skull called? That protects it to the pituitary gland. There's a special name for this area of the skull. Starts with an S. We talked about it a lot. The sella.
Cursic.
Yeah, perfect.
Yeah.
Sella turcica. Okay, that's the bony part of the sphenoid bone that comes out and surrounds the pituitary gland. Now, this is pituitary gland. What's this little stalk called that comes up into the brain?
Infundibulum.
That's perfect. That's the infundibulum. And when it feeds in the brain, what area does it feed into? What's this area of the brain?
Cerebellum.
Not the cerebellum, but the cerebellum is a little bit distal because it controls muscle movement. But that is the hypothalamus. The hypothalamus connects to the pituitary gland through the infundibulum. Like my great pictures. Looks just like it. So the infundibulum is a stalk that connects your hypothalamus to your pituitary gland, and the sella turcica is the bone that surrounds it as it develops. Okay. And it shows development again, just like the other ones, an invagination of epithelial tissue. And remember, there's two lobes, an anterior lobe and a posterior lobe. Okay. What's the anterior lobe called? What's the anterior lobe of the pituitary gland called? Adenohypophyses. Okay. And it's also called the hypophysis gland. That's the adenohypophyses. And both of those release hormones. All right, formation of the neural tube. Some of the neural cells migrate to form the central nervous system and form an aggregate that becomes your suprarenal glands. So the suprarenal glands actually start to form before the kidneys start to form. Okay, suprarenal gland and adrenal glands are the same thing. About week 5, they start to form the suprarenal glands. Okay, then the medulla forms first, then the cortex forms around it as the tissue pinches off inside. Remember, medulla is inside, cortex is outside for any gland. Okay, heart begins as a pair of tubes in the pharynx area. At week 3, the heart begins to pump. Okay, so the fetus has a heartbeat at week 3. Okay, the tubes fuse, forming a one-chambered heart. Starts off as a one-chambered heart. How many chambers are going to develop eventually?
4.
4. 4 chambers: the right atrium, right ventricle, left atrium, left ventricle. Okay. 2 large veins bring blood to the heart. Those are the umbilical veins. Are the umbilical veins oxygenated or deoxygenated blood? What do they have in them?
Deoxygenated.
Other one. Did you say oxygenated or deoxygenated?
I said deoxygenated.
Okay. They carry oxygenated blood, remember? This is the one place that's like, because it's carrying blood from the mother to the heart. So that's oxygenated blood. It's blood that mother has breathed in, got oxygenated. Okay, it's going towards the heart. Remember, that's why it's a vein. Then when it pumps it through the body, back out the umbilical cord, that's the umbilical artery. There's only one of those. Okay, and that has deoxygenated blood because the fetus has used that up. It's taking the oxygen out of the blood so it can grow, it can nourish itself, and then it's giving off carbon dioxide as a waste product and going back out through the umbilical artery. Because by definition, arteries go away from the heart, veins go towards the heart. And you got to remember the fetal circulation, you got the fetal heart now. So the blood coming in is coming from the mother, and that's oxygenated. Blood going out from the fetus is coming from the fetus back out towards the mother. That's deoxygenated. Okay. So you have 1 large artery and 2 veins. Your umbilical artery and 2 umbilical veins. The large artery carries blood away from the heart, and this is called the truncus arteriosus. Okay, here's your heart that starts to form. It starts from as 1 chamber. You see it has the 1 chamber at week 3, and it starts to beat with just that 1 chamber. As blood comes in, it beats through that and pumps blood back out. Okay, so at week 3, you have a heartbeat. 4 to 5, interarterial, interventricular septa begin to form. You start to take that 1 chamber, now you start to divide it. Remember, septa is the dividing wall. Interarterial means you're forming 2 atriums. Interventricular, you're forming 2 ventricles. So you're starting to form your 4 chambers at week 4 to 5. Okay, the foramen ovale and the ductus arteriosum form. The foramen ovale shunts blood from the right atrium to the left atrium, bypassing the pulmonary trunk. The ductus arteriosus shunts blood from the pulmonary trunk back into the left atrium. So both those are functioning to prevent blood from going to the lungs of the fetus, because it does no good to go there. because the lungs are not breathing in oxygen. The oxygen is strictly coming through the umbilical cord, the umbilical vein from the mom. Does that make sense? So those are your two shunts that are preventing the blood from going to the lungs of the fetus as the fetus develops. Now at birth, both these vents, both these shunts slam shut. They instantly close. They're supposed to instantly close. Sometimes they don't. That's where you get blue babies and things like that, because the blood's not going to the lungs for oxygenation. But if things work correctly, both the foramen ovale and the ductus arteriosus slam shut right at birth. Okay, so they both close at birth. The remnant is the fossa ovalis. That's what that's called. The closed portion is that little tissue. It's called the fossa ovalis from the foramen ovale, and the ductus ligamentum is from the ductus arteriosum. Okay? Those are the remnants that are on the heart, the tissue that's there where those shunts have slammed shut. Okay, and you can see the development of the heart from the tubes. You're starting to form your interatrial septum to interventricular systems, and now you're starting to form your four chambers of the heart. And you can see the ventricles becoming larger, and the atrium is starting to form on top. Okay, now you've got your interventricular septum. Here's your foramen ovale. Okay, you got when you're ready, you got the full four chambers of the heart formed. Right atrium, right ventricle, left atrium, left ventricle. The cardiovascular system. At week 4, you start to form the aortic arches. Remember the aortic arch? It starts to fuse together from the dorsal aorta. Okay, to form that in the fetus. Some of the aortic arches disintegrate, leaving only just one aortic arch. So as your aortic arch comes across, there's several of them, and the other ones disintegrate. So at birth, you only have the one aortic arch coming out from the left ventricle. So left ventricle comes up into the ascending aorta, your aortic arch, to your descending aorta. Your abdominal aorta goes through the diaphragm. Thoracic aorta, goes through the diaphragm to the abdominal aorta. Okay, so you have several aortic arches that are forming, and but the other ones disintegrate, leaving just one. Okay, so here's your aortic arches where they're developing. And again, these are going to disintegrate and leave just one at birth. Okay, you see how these are starting to disintegrate here. And again, then you just got this one aortic arch. Okay, remember there's the brachiocephalic trunk, which is the first one. The left common carotid and the left subclavian artery are the three branches that come off the aortic arch before it descends to the rest of the body. The vena cava, you have the superior vena cava and you have the inferior vena cava. Okay, you have your that drain back into the heart, and they both drain into the right atrium, both the superior and inferior vena cava. Okay, venous circulation. Vessels drain to the tissues from your anterior cardinal veins, your posterior cardinal veins, and your subcardinal veins. Okay, the fusion of the pair vessels form your superior vena cava and your inferior vena cava. Again, both of those dump into the right atrium. A true-false question most students miss on your boards will say the superior vena cava dumps blood into the right atrium, and the inferior vena cava dumps blood into the left atrium. That's a false statement. Okay, both these veins dump into the right atrium. Okay? And then blood is pushed out. Okay, here's your anterior cardinal veins forming from the heart, your posterior cardinal veins. These are eventually going to develop into the superior vena cava and your inferior vena cava. Okay. Again, here's the inferior vena cava coming up into the right atrium, the superior vena cava coming up down into the right atrium. Okay. Hepatic portal for your liver and umbilical vessels. In week 4, umbilical arteries carry blood to the placenta. So now the baby's starting to get its nourishment from the placenta, from the mother. Umbilical veins carry blood to the liver. Okay. So the arteries from the heart go back in, the veins carry back to the baby's liver and start to infill, and it can provide nourishment that way to the fetus. The right umbilical vein disintegrates about week 12. The ductus venosus allows some blood to bypass the liver. Again, the mother's liver is doing the work for the infant, for the fetus. Okay. Veins draining the digestive tract fuse to form the hepatic portal vein for the liver. Okay. At full term, before birth, blood travels to the liver and then to the inferior vena cava. So much of the blood bypasses the lungs, like we talked about, by traveling through the ductus arteriosus and the foraminal valley. You don't want the blood going to the lungs until after birth, when they have air. So at birth, now when at birth, those two bypasses shut, the pulmonary vessels dilate, the foraminal valley closes, the ductus arteriosus forms a ligamentum arteriosum. They close, and the pulmonary vessels dilate. So now blood being pumped from the heart goes into the lungs, and the baby takes its first breath. Okay. Again, you see that digestive tract, you see the hepatic portal vein here developing, and the ductus venosus, heart, and then your liver. Okay, same thing. Here's your umbilical veins. You have 2 umbilical arteries, and the umbilical vein. That's backwards. That's at full term. That's when they switch back over. When you switch, oops, don't pencils around. Okay. Okay, so at birth, everything switches back, and you get the placenta comes out. These veins fuse back together. Then you get your circulatory system of the newborn going as it should, nourishing the body, using its lungs, using its liver, and the body starts to function as you cut off the umbilical cord here. The lymphatic system gets 60 weeks. Thymus glands forms your pharyngeal pouches. Okay? The cells attach just inferior to the thyroid gland. You have the formation of your jugular lymph sacs, primordial lymph sacs, median lymph sacs. We call the cisternae chili. Remember, the cisternae chili is the starting point of your lymphatic system. Okay, the right lymphatic duct and the thoracic duct. The right lymphatic duct drains what? That's part of your lymphatic system. It drains the right head and neck, the right side above the diaphragm. Left lymphatic duct drains the entire left side plus the right side below the diaphragm. Okay, that's your lymphatic drainage right there. And lymph nodes occur throughout the body, numerous in places where infections can occur, like digestive tract, armpits, mammary glands, things like that. And there's numerous lymphatic tracts throughout the body are formed. So here's your medial lymphatic tract. That's going to form your cisternae chili. That's the beginning of your lymphatic system. The primordial lymph sacs and the jugular lymph sacs as they develop. Okay. Okay, the thymus gland, you're developing. Again, here's your parathyroid gland that's starting to develop. Your pharynx, and here's your thyroid. Thyroid has a right and left side with the isthmus that connects them. Okay, and behind it is your parathyroid glands, which you can see developing right back in here. And as these develop, you see the thymus starts to develop and your thyroid starts to develop on top of the thymus. Now the thymus is responsible for what? Thymus gland is responsible for maturing your T cells. Remember, B cells and T cells both develop in the bone marrow. They're both formed there. But the B cells become immunocompetent in the bone marrow, but the T cells do not. The T cells have to travel from the bone marrow to the thymus. In the thymus, then your T cells become immunocompetent. That's where they develop. Okay? But they're actually formed, they're not formed in the thymus, they're formed in the bone marrow. And they travel to the thymus to become immunocompetent or activated. Okay, and again, here's the cisternae chili developing from the median one here. This is the beginning of your lymphatic duct, the thoracic duct. Then the thoracic duct runs up, and then the right lymphatic duct. Okay, that's the drainage for your system right there. You have lymph nodes develop. You have a medulla and your cortex. You have a capsule surrounding them, and then arterial supplies in the lymph nodes, and it filters out the bloodstream, and it collects pathogens, bacteria, things like that that aren't supposed to be there. That's where they swell up. All right, let's do respiratory system, and then we'll take a 10-minute break. How much longer do we have on this, by the way? We have a lot more slides.
44 slides,
We have what?
43 slides,
3?
43.
Oh, okay. We'll do this, then we'll take a break in about a couple minutes and do about 10-minute break. All right, so the respiratory system. Respiratory has a pulmonary groove, which forms your pharynx area. The groove forms deep, but it's 2 blind pouches at the end of the groove. Those blind pouches are going to become your lungs. Okay. The grooves form a tube, which becomes your trachea. The pouches become the lungs. And the diaphragm starts to form about week 9. Okay. At 3 months, the lung bud is going to be branched into numerous tissues. And by 6 months, there's millions of branches. Okay. The bronchioles are formed, and all the alveoli, which are the little sacs at the end of the bronchial tubes, that where gas exchange occurs when you breathe in and out. Okay. So at week 3, you see the pulmonary groove. At 4 weeks, you see the pulmonary groove starts to develop, and you have these 2 lung buds develop. The blind sacs are going to form your lungs. This tube here is going to form your trachea. Okay. You see the lung buds start to develop, become more and more branched, more and more branched. At the ends of each one of these branches, you know, these little cul-de-sacs here of alveoli. And they're very thin, a huge blood supply surrounding them. That's where the gas exchange occurs, at the alveoli. Very thin epithelium there. Pleural cavities. The pleural cavities are what contains the lungs. Okay, you see they're going to form. You have your digestive, the lung buds. You see the pleural cavities are starting to form around it. Okay, again, you have your developing lungs, week 6. Week 8, you can see the pleural cavities. Remember, the lungs are an organ that forms within an epithelium. So it's like that balloon we always talk about. You take your fist and push it in a balloon. There's 2 layers. That's what the pleural cavity is. It has an inner layer, which is your visceral, which actually touches the lungs. That's this side of the pleural cavity. So week 3, excuse me. Okay, the yolk sac is formed. So the layer between the yolk sac and the amniotic sac develops your hindgut and your foregut. The hindgut and foregut form the digestive tube. So lateral to digestive tube on the sides of it are cavities. Those are called the colemic cavities. Digestive tube is held in position with the midline of the colon cavities by the dorsal mesentery and the ventral mesentery. Okay. At week 4, the pancreas is formed within the dorsal mesentery area. The liver is formed within the ventral mesentery area. Part of the ventral mesentery becomes the falciform ligament of the liver that holds it in place. The remaining mesentery tube becomes the greater omentum and the lesser omentum. Remember, the greater omentum attaches to the greater curvature of the stomach. The lesser omentum attaches to the lesser curvature of the stomach. The lesser omentum comes from the lesser curvature of the stomach to the liver. The greater omentum comes from the greater curvature of the liver all the way down to cover most of the organs in the peritoneal cavity. The intestines elongate, and then they start to coil at about week 6. The yolk sac and the body stalk that are developed, those 2 form and start to form the umbilical cord. Okay. So here's your yolk sac. This is going to form the umbilical cord here. And this is your hindgut that starts to form. And the somatic tissue around the neurocele here, around the nervous system. And this is your developmental and your mesoderm. And this is the hindgut that's starting to form your the tube of the digestive cavity. Okay. You got your digestive tube starting to form. And this is your colonic cavity with the ventral mesentery starting to form the ventral surface and the dorsal mesentery that's going to form on the dorsal surface. And as time progresses on, you can watch how the development of the liver as it starts to invaginate from the different areas, and pancreas starts to form two separate organs. And there's your liver and pancreas. Okay? As everything differentiates and invaginates as you embryologically progress. Okay. So at week 6, you have a pancreas, and you have the umbilical stalk that fuses with the yolk sac to form your umbilical cord. You have a liver, you have a stomach, you can see the gallbladder starting to form there also. So at 8 weeks, you're starting to have the ends of the trachea starting to develop. Right? You have your esophagus that's formed. Remember, the trachea is anterior to your esophagus. It's in front of it. Okay, that's we have the epiglottis rolls over the glottis to prevent food entering here to fall under the trachea. It has to pass over it. You can see the development of the liver. Okay, you can see the stomach. You see the pancreas, the gallbladder, and the intestines. The small and large intestines are just starting to develop and starting to coil around. That's at 8 weeks. Okay. At 10 weeks, okay, you have a defined heart, defined stomach, liver, gallbladder, and the intestines are a great deal coiled. And you're starting to get differentiation between the small intestines and the large intestines.